The last functional group that we're going to be talking about for organic acids and bases are going to be amides. And so what an amide looks like is it's going to be a carbonyl next to an amine. And so you can have monosubstituted or disubstituted amides. This would be a monosubstituted, and this would be a disubstituted amide. R1, R2. And so can you guys see why one is monosubstituted and one is disubstituted? Yep, it's the R groups, right? So you know amines, we have classifications for amines. We have primary, secondary, or tertiary, which is related to the number of alkyl groups that are bonded to that amine, right? So with amides, we either have one R group or two R groups. And that's because nitrogen likes to form 3 bonds if it's neutral. And when we have an amide, we have one spot that's already taken up by the what we call the acyl group, that R double bond carbonyl-carbon oxygen, right? So one spot is taken up by the blue group there, which is the acyl group or the carboxyl group, and the other positions are either filled with hydrogen or nitrogen. And so we can also have a not substituted amide as well, where you just have all nitrogens there. And so this is an amine side plus a carboxylic acid side. And amides are extremely important functional groups because these form the backbone. Does anyone know what these form the backbone for? Anyone taking any biology classes? Amino acids, and if you put a bunch of amino acids together, you get a protein, right? So this forms the protein backbone. All right, amide functional groups form the protein backbone. And some other information about amides is the N-C double bond. Here, this bond has slight double bond character. And the only reason that's important is because when we have a protein, it means that rotation about this bond, the amide bond, is slightly restricted. Question?
You have it's like a nitrogen?
Yeah, this is part of the amide right here. So there are 2 R groups, maybe 3 R groups here, and then this is a dashed line. Right, it's just saying that bond, whenever you see that dashed bond, we showed a little bit during our discussion on Lewis structures, represents a resonance between the 2, so this bond alternates. This bond is slightly double bond in character, which means that it's kind of hard to rotate it. It isn't, something that will affect your guys' understanding of amides very much, but in terms of proteins, it's important because this restricts the rotation of the protein backbone. And what we'll learn is that with proteins, structure and function go hand in hand. If you break the structure, you break the function of the protein or enzyme. And the naming, again, I'm not going to be testing you guys explicitly on the naming of amides, but the way the amides are named is you put N, then the alphabetical order of alkyl groups, and then to that you add the carboxylic acid group plus amide. If there's only one group on the amide, you don't have to put NN, right? You would put N-methyl, for example, N-methyl hexane amide. You might also see carboamide, if you're studying with other sources and stuff. Carboamide is just replacing the amide there. It's in specific scenarios that you use carboamide. But like I said, I'm not gonna be testing you guys on the naming of amides, just the, looks like this got loaded again. Here, let me get that out. Just what is the functional group, identifying the functional group, and realizing this is important for protein structure. So what do you think for amides, if you have an amide, what's the strongest intermolecular force that you'll have just by looking at this structure? Hydrogen bonding, right? I know it's early, right? So we have a nitrogen bonded to a hydrogen, which means this is going to be capable of hydrogen bonding. This nitrogen can act as both a hydrogen bond acceptor and donor, and then this carbonyl group right here can act as a hydrogen bond acceptor, right? And so I'm just introducing the idea that in proteins, secondary structure of proteins is held together by these hydrogen bonds that are formed between the amide functional groups on the backbone. And just to give you an example of what an amide name is, like I said, I'm not going to be testing, so I'm not testing on naming. Let me put that up here in red for everyone. Not tested on naming, but just so you can get an idea of what this looks like. Am I good to go on to the next slide, or is anyone still writing? This says carboamide. So for example, if you have an amide on a cyclohexane, it's not part of the parent chain of the cyclohexane, so you treat that as an alkyl group, and so it ends up being, because if it was a carboxylic acid, it gets called cyclohexane carboxylic acid. So if it's an amide instead of a carboxylic acid, it's called cyclohexane carboamide. Trust me, the names, I'm testing my 212 students on naming carboxylic acid derivatives, esters, all these things, and to be frank with you guys, the naming just doesn't, I don't like it. I don't like it. Once you get to a certain complexity of functional groups, the common names are incorporated into the IUPAC names. And so one molecule can have 4 different names. Okay, so let's look at just a quick example. So here we have an amide, and it's going to be a simple amide, 4, 5. And then this is an H here. We see that we have one group on this amide right here. It's just a methyl group. And so we're going to put N-methyl because it's on the amide. And then we name the carboxylic acid side of the chain, and we add amide. So then there's a 1, 2, 3, 4, 5 carbons there. So this becomes N-methyl pentanamide. And that's because it's an amide molecule. That's the functional group. All right, so that might be a name that you would see for an amide. Like I said, I'm not testing you guys on naming amides. So this is the N-methyl, and this is the pentanamide. Okay.
And the methyl for the one carbon coming out of the nitrogen group?
Yeah, it's just the methyl group right here. So this is the carbon skeleton structure for just a methyl group, right? This is a CH3 group right here, and this is the bond to the nitrogen. The nitrogen also has one lone pair there, right? So remember, whenever we have heteroatoms, meaning not carbon or hydrogen, the line to the heteroatom represents a bond from carbon to that atom. There is no extra carbon here, right? There's only one carbon here. There's no carbon there. Okay. And so the formation of amide is similar to the formation of esters. I don't know why there's a question mark there. Oh, it says, what nitrogen compound to a carboxylic acid forms an amide? Remember when esters, we described the molecule in terms of having an alcohol side, and a carboxylic acid side. With an amide, we have a carboxylic acid side, and what do you think the other side is gonna be? What, the amine side, right? So you have the amino side and the carboxylic acid side. And so we add an amino side. Let me make that green because nitrogen is green in my head. So we have an amino side and a carboxylic acid side of the amide. And so generally, all you have to do to make an amide is you mix a carboxylic acid with an amine. The amine can only go up to a secondary amine because you need that hydrogen, at least one hydrogen there, to go through the reaction mechanism to form the amide, right? So at most, it can be a secondary amine, and generally this is going to require a catalyst and acidic conditions, right? So if you know biology, you know that proteins are synthesized by ribosomes, which basically they lower the activation energy, right? So this is a very unfavorable reaction, which means that we need to add some energy into this reaction to get it to form an amide molecule. And so all we do is the exact same thing we did. Sorry, I know it really feels like it's flickering a lot today. Is we do the same thing with the ester, where we took the alcohol side and the amine side, or sorry, the carboxylic acid side, and you combine them together to form the amide functional group, right? So we're gonna take, and I said this proton is important because you need it for the amide reaction to happen, and this nitrogen is going to bond to this carbonyl, and we're gonna lose the OH and lose the H here, right? So what this is going to look like is we will have R carbonyl, and then the amino side right here. And if we count oxygens and hydrogens on the left-hand side of this reaction and on the right-hand side of this reaction, what can you say? How many carbons or oxygens and hydrogens did I explicitly draw on the left-hand side? Two hydrogens, and how many oxygens? Two, right? And how many do I have on the product side of this reaction? We have one oxygen, which means you lost one oxygen, and what else do we lose? Hydrogen. How many hydrogens do we lose? Two. So what do you guys think the byproduct of this reaction is gonna be? Water, right? So this is gonna be exactly the same as esterification, right? Where we get the byproduct of water from an alcohol and a carboxylic acid. An amide works the same way, except we have an amine instead of an alcohol. And we can only go up to, this only works for primary and secondary amines. Right, so anything, if you have a tertiary amine, the reaction will not happen. The nitrogen-carbon bond is too hard to break. I'm sure there are specific reaction conditions where you could eventually get that to happen, but we're not worried about those. Okay, am I good to start an example of this, right? So for this, predict, bless you, the products for this reaction. We have, and we're not going to name anything here, OH plus in the presence of acid. Right, so where I had the R groups, identify that in this reaction, and then just put the new R groups with the appropriate actual group when you're predicting the products. I don't know why I did everything. I will also write.
Is that like the second one?
Yeah, that's the second one. Yep. All right. So what did you guys get for your first product? What does it look like?
Just like that, together.
Just like that, but together. Exactly right. So whenever we're doing these condensation reactions, that's the basic idea. We just have our reactions, and we're just putting them together in one way. We need one hydrogen to get rid of on the amine, at least, and we're going to get rid of the OH on the carboxylic acid because what's a byproduct? Water, right? So we need to lose those. So we're going to take this nitrogen, and it's going to bond to the carbonyl carbon, right? And we're going to lose the hydrogen and the hydroxide groups. So this is just going to look like that, right? Here is the amide bond, and the byproduct, like you guys said, is going to be water. Is that what everyone got for their amide?
Would we be wrong in drawing the H2O?
In drawing it? Oh, you mean if you just drew that?
Yeah.
No, not at all. Yeah, I like drawing the structure more than I like the chemical formula. Ethan?
If you wrote like R1 and R2, is that fine?
So for this one, because it's specifically asking, so that's a good question. For predict the products, we have a general reaction, right? So this, what I have here is a general reaction for this because R1 and R2 are arbitrary. When I'm asking for predicts the products, I'm asking for specifically these molecules, right? So you don't put R1 or R2. R1 and R2 are good to have on, so for the final, you'll have a full sheet of paper. R1 and R2 will be good for your cheat sheet, right? Because then you know the general mechanism or the general product for general reactants, and you can apply that to whichever specific molecules you're given. So go ahead.
This one is it would be a carbon ending it with hydrogens, is that?
Carbon, yeah, so this is two methyl groups that are on the amine. So for example, if we had to classify this amine, we go all the way back here, this would be dimethylamine is what it would be called. And then for the second one, and for this amide that we just drew, is this mono or disubstituted? Right, how many R groups do we have on the nitrogen in the amide? Two, so it'll be disubstituted, right? The number of R groups is the kind of how substituted the amide is. So this is a disubstituted amide. And then for the bottom one, it's the exact same idea. We're gonna lose the hydrogen and the hydroxyl group, and the nitrogen is going to bind to the carbonyl carbon. So I'm gonna draw that. This is the carboxylic acid. Now it's gonna bind to the amide, and then we have an ethyl group coming out of there. And what's in the other spot for the amide? Right, so if we look at this one right here, we have, what is this a primary or secondary amine?
That is the
Primary, right? Because we have one R group, right? So it's a primary amine, which means that one of the spots is going to have to be filled by the other hydrogen, right? And this is also going to have water as a byproduct because that's always a byproduct. And is this mono or disubstituted?
mono.
This will be mono because we only have one R group on the possible two positions for the amino side of the amide. So this is mono substituted.
What if we drew it like how having the react we put like the group, the R group, like that
Mhm.
with hydrogen like that.
So if you drew it going down instead?
Yeah.
Yeah, if you drew it going down instead, that's okay. I wouldn't mark you down for that. Right, so Maya's question was, if we drew this like that instead, is that all right? Yeah, that's still fine. Technically, it would probably be this way. It's actually the actual structure because of that restriction of the amide bond, but that would be fine for predicting the product here. Okay. How do you guys feel about that? Thumbs up, thumbs to the side, thumbs down? Do you guys want another example, or you want to move on to hydrolysis? I heard some mumbles. Okay, so let's go on to hydrolysis. And so the same thing with esters, right? We had the esterification reaction, which is a condensation reaction, and then hydrolysis was just the opposite of that. And so the hydrolysis of amide is the exact same thing as the hydrolysis of an ester, except instead of a carboxylate and an alcohol, we end up with a carboxylic acid and a, what do you think the other product is going to be? Huh? That's for an ester, right? Ester is carboxylic acid and alcohol. An amide is going to be a carboxylic acid and amine, right? So it's the same idea. If we can identify the amino side and the carboxylic acid side, whenever we do the condensation or the hydrolysis, it just goes in opposite directions. And so this needs a catalyst to happen. And it could be acid catalyzed or base catalyzed, this reaction. So we'll just stick with the catalyst. And so we need a carboxylic acid and an amine as our products, right? Because for an amide, we have our R1, R2, our amino side, and we have our carboxylic acid side, right? And so if we can identify this, which is always going to be our starting molecule in hydrolysis, you've done all the work for the problem. And so the general reaction is going to be R double bond O N R1 R2 H3O plus, and maybe some other type of catalyst like an enzyme. And we will get ROH plus NHR1 R2. So this is our amine, and this is our carboxylic acid. And so looking at the hydrogens on the reactant side and the product side, what else do you think is going to be needed for this reaction to happen? Water, right? So water is going to have to be consumed because we need to add a hydroxyl group to the carboxylic acid, and we need to add a hydrogen to the amine group. Right? So we need plus H2O. An example of this, and again, the idea is exactly the same. Sorry, excuse me, I'm getting like a lot of hairs in my throat or something. Exactly the same as the condensation, the formation of an amide, except we are breaking an amide apart, but we're still doing the same type of identification for these reactions. H3O plus, so this is again predict the products. And I will give you another one. So let's first, for both these reactions, identify the amine and the carboxylic acid side. So the amine side for the first molecule is going to be this, and on the second molecule, it's going to be that part. The carboxylic acid side has the carbonyl. So this is going to be the carboxylic acid side for both molecules. Right, and so for both of these, we're just gonna draw what's I've circled here and then adjust them to make them either an amine or a carboxylic acid. Doesn't matter how many substituent groups are on there, I will give you reactions where you don't have potential side reactions going on, right? So you don't ever have to worry about that. All right, so those are our carboxylic acid sides, what I've circled in blue there. And what do I need to add to both of these? OH, right? Because carboxylic acid has an OH group. All right, so those are both of our carboxylic acids. And then I'm going to draw what I have for the amines. So what I have circled in green here and H. And what do I need to add to both of these? An H, right? We need to add a hydrogen to then complete the octet for nitrogen, and those are our amine sides. And this would be the products for this reaction. And so these ones, I won't ask you to name the products, at least for not the second one. The first one, though, I could ask you to name both the carboxylic acid and the amine, right? So what would be the name of the carboxylic acid here? Yeah, for the first one. For the first one. No, the cyclo is only for cyclic molecules, right? We have 1, 2, 3, 4 carbons, right? And then what groups do we have there? We have methyl groups, and how many of them do we have? And they're on which carbons? 2 and 3, right? So we have 2, 3, dimethyl. And what's the parent chain name for 4? Bute. Are there any double bonds? Nope, so we just put butane. And then what's the ending for carboxylic acid? It has acid in it. So you just have to find, if we think of it an acid, think about how the name changes, right? So HCl, it's oic acid, right? We always have an ic ending at the end of acids. Usually oic, if it's an oxy acid, so it has oxygen as part of it, right? So it's butanoic acid. That's how we would name that compound. And the amine, how would we name the amine? What groups do we have on the amine? For the black star right there.
Oh, yeah. Nitrogen.
We have 2 methyl groups, right? And remember, amines are easy. We just do the equivalent of a run-on sentence in the name, where we just list all the groups in alphabetical order and we just add amine to the end of it.
And methyl.
Yeah, so you guys aren't gonna have to put the N just for the amines, but it'll be dimethylamine is what we'll call this. So this will be dimethyl amine. And then how would you classify that amine? Primary, secondary, or tertiary? Secondary, because we have 2 groups on it, right? This is a secondary amine. All right, so same thing when we're putting this is just the opposite when we're putting 2 molecules together. When we're breaking them apart, we're going to identify the amino side, the carboxylic acid side, and that's just going to be the products for what we have. The same thing when we have an ester and we do the exact same thing. For the first one, that carboxylic acid, you're not going to have to know how to name something like that. That would be called cyclopentane carboxylic acid. And the second one, what you guys could name that amine. What would the amine name be? What is it? Primary, secondary, or tertiary? Primary, right? Because we only have 1 group, right? There's only 1 R group. All the other groups are nitrogens.
So that's not 3, it's just 1 because the R gonna end at the end of the thing?
Say that again.
So like, you know how I see there's 3?
Mhm.
Like 1, 2, 3, or whatever?
Here, this.
Mhm.
This is just 1 alkyl group that's bonded to the amine here. Yeah. So it's the same thing if you had something like this and you just put the isopropyl group there. Oh, dang. And so what's that group called? Isopropyl, right? So this It doesn't have to do with the number of carbons that are bonded to it, it's the number of carbon groups that are bonded to it. This only has one spot on the possible 3 spots for the amine. Right? The other spots are filled with hydrogen, so it doesn't count towards the classification of the amine. You can think of it in 2 ways. If there's 2 hydrogens bonded to the amine, then it's going to be a primary amine. If there's 1 hydrogen bonded to it, it's going to be a secondary amine. If there's no hydrogens, it's going to be tertiary. So hydrogens and the alkyl groups follow the opposite trend. Right? So you can think in terms of the number of hydrogens there or the number of R groups that are there. Not the number of carbons in the R group, but just the number of R groups. Morning. And so what would the name for this be then? Right? What was this group called? Isopropyl. And are there any other groups? Nope, so we just call it isopropylamine. That's how we would name that structure. Good. All right, so that'll wrap it up for organic acids and bases. This is a pretty, I would say, fundamental lecture for the rest of the lectures because organic acids and bases make up the majority of functional groups in biomolecules. So carbohydrates have aldehydes and ketones. Proteins have amides, carboxylic acids, and amines. Lipids have carboxylic acids, alcohols, amines, carbohydrates involved, and lipids are really complicated. And then nucleic acids, those will have a little bit of everything and other more complicated functional groups. Right, but for all the biomolecules, yeah, so the carbohydrates are aldehydes and ketones. I'll make a little table right here. So the biomolecule functional groups, and this will be a simplified version because you can always modify these biomolecules. Functional groups as a prelude to what we'll be talking about for the rest of the class. Carbohydrates have aldehydes plus ketones and alcohol groups in there as well, but they're classified by aldehydes or ketones. Proteins have amines, amides, and carboxylic acids. Lipids are not classified by a functional group, but the functional groups you will see there are going to be carboxylic acids, alcohols, amines, out of the functional groups that we've talked about so far. And then nucleic acids will be a little bit of everything kind of in there. It's mostly going to be amines, amides, carbonyl functional groups, but the really ones that kind of classify that biomolecule are for proteins and ketones. Lipids are more classified by their physical properties, and nucleic acids are very complicated molecules that I wouldn't put under these functional groups that we've talked about so far. But the chemistry that's involved with these molecules is relevant to the organic acids and bases. Why? Because acids and bases are very reactive, and your biomolecules are very reactive. You need to go through these reactions for a lot of important biological processes. Okay.